My name is Margaret McCormick, and I recently graduated with a master's degree from the Mitten Lab in the Ecology and Evolutionary Biology Department at the University of Colorado at Boulder.
My master's research focused on the genetic variation with elevation in yellow stonecrop,
a common plant that's found at an unusually wide range of elevations here in the Rockies.
One of the most striking features of the mountains is how the vegetation changes as you climb from low to high elevation. The grasses and wildflowers common on the plains become sparser as you begin to ascend, and you start to see conifers of various kinds. On the lower slopes, ponderosa pine and blue spruce are common, while higher up you are more likely to find lodgepole pines, Engelmann spruces, and subalpine fir. Higher still, trees become stunted, then scarce, giving way to diminutive alpine grasses and herbs distinct from those of the plains. Thus most species have a specific range of elevations in which they thrive, with similar but distinct species occupying different zones. These zones reflect genetic differences between the species. Each is adapted to the particular conditions, both in climate and other organisms, encountered where it is found. Yellow stonecrop, scientific name Sedum lanceolatum, appears to buck this trend. This small succulent wildflower is common in the Rocky Mountains and indeed across the western portion of North America all the way to the coast. Its elevation range is greatest in Colorado. I have found it growing at or below 6,000 feet near Boulder, Colorado, all the way up past 12,000 feet along the Continental Divide. A subspecies has been described in alpine habitats, but as far as we know it is the same species at all elevations. Genetic variation in yellow stonecrop has been examined across the entire Rocky Mountain range, but not at a local geographic scale nor specifically with elevation. I therefore set out to investigate what kind of genetic differences might exist between stonecrop plants at different elevations. To do this, I sampled a number of plants from over 30 populations across the range of elevations between Boulder, Colorado, and the top of Niwot Ridge, as shown on the map. I collected a small sprig from each plant, extracted the DNA, and amplified and sequenced individual genes.
I found an astonishing amount of genetic variation across the different populations in both locations. In fact, some of the genes had so much variation that it was impractical to analyze with the methods I used. But the most striking variation I found was in a chloroplast gene called matrase K.
In both plants and animals, most of the genetic material resides in the nucleus of each cell. This DNA follows the typical rules of inheritance. An individual gets half from the mother and half from the father, and might pass on the maternal or paternal copy of any given gene to the next generation. But there is also DNA in certain structures within eukaryotic cells, the mitochondria in animals, and both mitochondria and chloroplasts in plants. The DNA in these organelles, as they are called, is almost exclusively inherited from only one parent, the mother in most species.
In animals, mitochondrial DNA has a much higher mutation rate than nuclear DNA, and so mitochondrial DNA is routinely included in studies of genetic variation in animals because there's a lot to find. But the opposite is true in plants. Chloroplast DNA mutates about a third as fast as nuclear DNA, and mitochondrial DNA mutates about half as fast as that. This is especially evident when you look at protein coding sections of genes. Mutations in these regions have the potential to alter the protein that is made, which can then affect how it functions in the cell. Sickle cell anemia is a drastic example of this, and is caused by a single protein-changing mutation in hemoglobin. The function of both mitochondria and chloroplast are thought to be so fine-tuned in plants that protein-changing mutations are rarely ever tolerated. Therefore, plant geneticists have often assumed that there is little variation in organellar DNA to be found between individuals of the same species, and none at all in protein-coding sequences. My data suggest otherwise for yellow stonecrop.
I collected sequence data for 2 genes in mitochondrial DNA and 3 genes in chloroplast DNA. The 2 mitochondrial genes, COX-2 and ATP1, both encode components of the electron transport chain, which converts sugars into usable energy for the cell. This graph shows the approximate longitude and elevation of the individuals I sequenced, with different colors representing the 4 different versions I found for both genes. This is incredibly high genetic diversity for plant mitochondria, especially since these plants are at most 50 miles apart. However, one version is found everywhere, and I did not find any protein-changing variation in either gene. Overall, these data suggest that mitochondrial protein differences may be less important for stonecrop's adaptation to different elevations. The story gets more intriguing in the chloroplast. Here I examined two adjacent genes that code for parts of the chloroplast ribosomes, which are the structures that assemble proteins according to genetic instructions, and MATK, a matrase enzyme which helps process the RNA instructions for use by the ribosomes. All three are critical to protein synthesis in the chloroplast, and therefore to the chloroplast function as a whole. Ribosomal proteins in particular are remarkably stable in all living things. For example, most are recognizably similar between bacteria and humans. I found 4 versions of the ribosomal genes combined, again represented by different colors on the graph. Unlike the mitochondrial genes, here the proteins that are encoded are actually different between all 4 versions. Although the minority versions are rare compared to the most common version, this represents an incredible wealth of genetic diversity for chloroplast DNA and ribosomal genes in particular, especially with the differences in the proteins themselves. But the really astonishing variation was in MATK. I found at least 18 different versions of the protein encoded in this gene, and there are more versions if you count ones that don't change the protein. MATK is a notoriously fast mutating gene, but the amount of variation I found, within such a small geographic range, is unheard of. It's on par with the differences found between different species. Most of the protein versions are also not evenly distributed with elevation. For example, the red variants are found mostly at lower elevations, while the blue variants are mostly at high elevations.
It's possible that the different variants arose at certain elevations and simply haven't spread to other areas yet. Or, intriguingly, it is possible that certain variants are actually advantageous where they are more common. While my data alone can't distinguish between these two possibilities, it is clear that yellow stonecrop harbors an unexpected amount of genetic variation, and that mitochondrial and chloroplast DNA may play an underappreciated role in plants' adaptation to elevation.
This work was funded in part by the Indian Peaks Wilderness Alliance. Thank you very much for your support and interest in my research.